important stuff. So, right, when we look at each one of these tests, we should look at it via the scientific method, right? So, for the methyl red test, which were the methyl red VP test, so this was media that you had inoculated for both of these tests. And then we took samples out, right? And we aliquoted it 1 ml into one tube, 1 ml into another tube, right? So that we could run these two separate tests. One of them is the methyl red test using methyl red, which is a pH indicator, right? And it will stay red. It started out red. So if you guys are looking at your unknown reports right now, which I would look at as well for the methyl red test and review your results, right? It started out red. If we added it and that pH was 4.4 or lower, it stayed red, which means that your sample is very acidic. So it's positive for what we call mixed acid fermentation. That's what the methyl red test tests for, right? For the ability to produce lots of different acids that really lower the pH. Everyone with me on that one? Kind of, sort of, right? So it's a pH test. We read it immediately. If yours didn't have a low pH, what happened to the color of the methyl red? It turned yellow, right? So it was higher than 4.4, right? So you didn't have a lot of acid, so it was negative for this test. The other test we ran, which was the VP test, the Vaux-Proskauer, yeah, I'm all right. We used several reagents, alpha-nethaphenol, right? And then we mixed it and we added potassium hydroxide, and we really mixed it the second time after the second reagent because we were adding in what? We were adding in oxygen, right? Because oxygen is needed because this is a chemical reaction that's happening, right? And what are we reacting with? One of the intermediates in the pathway, acetone, right? Acetonin, excuse me. And we let this one go for, 60 minutes before we definitely recorded it as a negative. What color did you see if it was positive? Should you have seen? Red, right? So hopefully everyone's got your correct results. I'll check in with you guys once we get finished lecturing today and make sure that you guys have got what you should. So what do the results mean? So as we said, red, which forms immediately for the methyl red test because it's a pH indicator, right, is positive. And red is also the color, which happens to be the color that forms for the VP test. But this one we got to wait because it's a chemical reaction that's got to happen, right? So we wait at least an hour, right? Negative results for both ones are no color. And the reason for that, especially with the methyl red, it turns yellow, which is basically the same color as the media, so sometimes it almost seems like it disappears, right? It's kind of cool. It looks like an experiment. Sometimes you'll get a discolorization with the VP test, but if it's not a red color, a reddish color, it's not positive, okay? So only if you had a red color last time should you have recorded positive results. If you had other colors, you can write that, right, but don't consider it positive. So what does this mean? Again, what does this tell us about our organisms? So for the methyl red test, right, we're testing for a lot of acid. It means that yours is a mixed acid fermenter. When it ferments the glucose in this media, it produces a lot of acid.
And then drop the pH, right?
Right, drops the pH way low. Right, 4.4 or lower.
So if it's low, above 4.4, is no change, it's yellow, right?
Yeah. And it means that it's not producing a lot of acid. It still could be producing acid, and you guys already know this from your phenol red tests, right? Some of yours produced acid, just not to that level, right, where you have a lot of acid. Good questions. Okay, and for the VP test, although we're testing for the presence of acetonin, it's actually an intermediate to the final question that we're asking is, if they're producing acetonin, they can convert that all the way to 2,3-butanol. And what type of substance is butanol? An alcohol, right? It's an alcohol. So remember, what are the common products from fermentation? We get acids, which we're seeing here with the mixed acid test, right?
A lower pH.
Right, that acids affect pH. Gases and alcohols, right? And the VP test is actually the only one that we do that actually tests for an alcohol, right? All the rest we're dealing with acids. And one of them we're actually able to detect gas.
For the MR test.
And that is this one. So our Phenol Red test is the only one that can detect gas. And the reason for that is this little tube inside your tubes, right? What we call the derm tube. And it'll trap the gas, right, when the gas is produced. And so we see this bubble. We don't know which gas it is. It could be hydrogen gas. It could be carbon dioxide gas. We just know it's gas, right? But those are the two common gases they produce when they ferment.
Based on the color?
Yeah, I haven't gotten to that yet. So the color change, again, is because we've got a pH indicator. The name of the pH indicator is the name of the test, Phenol Red, right? So this one starts out kind of orangey-ish red when it's at neutral, okay? If you have growth of your organism, but no color change, right? It's not fermenting, it's not doing anything, okay? If it ferments, there's two possibilities. You get acid production, and we know this because it turns what color? Yellow, okay? If you get acid and gas, you've got yellow and bubbles in your derm tube, right? So make sure you draw those on your guys' report as well. Draw in your tube, draw in your bubble, right? Or draw in your tube and no bubble if you don't have a bubble. Now, some of us are getting a little tiny artifactual bubble. Not unless it's a significant size bubble do you record it as gas, okay? And so compare it to our control racks that I have for you guys. And if you have any question, let me know. Yours is definitely a bubble. I'm talking about like tiny bubbles, right? Don't be looking for little microscopic bubbles. If it's a significant bubble, you will see it. There's no question, okay? And so those are the two possibilities with fermentation, right? When we ferment sugars. But the other choice in this tube is there's protein source in this tube. So the organisms could use that for energy. And when they do that, they're going to produce alkaline or basic products. And so therefore that also has an effect on the pH. So in this case, we go to this end that we term it K for alkaline. And it's this pinkish magenta color, which again, you guys have the control tubes in your racks that you can compare it to. And so you know what that color and what that symbol means, right? K is for eating the proteins, the peptones, producing those alkaline products. So we are dealing with glucose for our tests. But there's lots of different sugars you can test for, right? But we've limited it to just one. So this is a multitude of different sugar and sugar alcohols that can be fermented by organisms that are available from the manufacturer that we use. Now the broth itself, with just the protein source and no carbohydrate added, can also be purchased, which we do not purchase. We purchase it already with the glucose in it. Some scientists would use just the base broth without any carbohydrates to it as a kind of a negative control test, okay? Which we didn't do, but we're going to talk about that. So notice the components in this, pancreatic digest of casein. Casein is a protein found in milk. It actually makes milk white. And so this has been partially digested for the bacteria. So this is the protein source. There's salt to maintain a osmotic balance, right? It's a normal isotonic solution. Glucose, as I said, is the sugar that we have. Phenol Red is what? The pH indicator, and then water, right? So that's all that's really in this media for those organisms, right? So really the only things for them to eat is either the protein or the sugar.
Glucose first,
Which,
preferably?
yeah. Preferably, most organisms going to eat the glucose first. And this is why we talked about last time why we only incubate them for a short period of time. And I keep forgetting hers in the incubator. And her organism is kind of like me, that it'll eat the sugar first and then it'll start eating the protein. But we want to catch it when it's eating the sugar. We want to see that it's eating the sugar and that it's not just choosing to eat the protein, okay? So I always use the little story of, you know, I've got the hamburger frying on the grill, but it takes a while to cook the protein. So in the meantime, I'm eating candy, my sugar, before my protein is ready. And a lot of organisms are like that. You know, they're gonna eat the sugar first. But there are some that don't ferment the sugars and go right to the protein. And we actually have 2 of them, and I think Shannon has 1 of them, which is why yours is pink. And it's supposed to be pink. And you'll also see that there's no gas bubble, where there's a little bit of a clue with Yvonne's, because you have a gas bubble, don't you? Do you have a gas bubble in yours or not? No gas bubble. Well, oh, I know who does. Muhammad, you have a gas bubble in yours, don't you? And his is actually orange right now because it's been, even though I put it in the fridge, I really couldn't stop his organism from starting to try, now that it's run out of sugar, to go to that protein. And that one will even start to do it after 48 hours. Like you'll get this little layer of orange on top of the yellow layer. So what is happening is they're eating the sugar, right? And especially his is a gas producer. So that's a clue right there. Like if you were to let it go, if I left it in the incubator like I did Yvonne's and it turned pink, that bubble would clue you in that, well, wait a minute, maybe something happened before he ate the protein. Maybe they digested or fermented the sugar first, because that's the only way you get gas production. And that's an important point, that it's only with fermentation that you're going to get gas production, right? So if you've got a pink tube with gas, then the question is, what's going on here, right? How did that happen? They had to have eaten the sugar in order to get that gas bubble. So that brings us to, so again, this is our carbon source, this is our protein source, our amino source. So again, we're looking at gases, not alcohols in this test, right? And we can look at acids as far as fermentation goes. And I want to fast forward to the table that I skipped over last time. So this is what I talked about right now, right? What if the bacteria eats the sugar and then began to eat the protein? So you're going to get that acid production, it's going to turn yellow, right? But as the ammonia is produced, your pH goes back up. And when you look at the pH scale, right, if we start out yellow, eventually we're going to go back orange, which is where his is at, right? And then if we put his in the incubator and let it keep going, eventually it's going to turn pink, right?
You just described something that I hadn't observed before, but you said every now and then it's yellow at the bottom, and at the top has a little pink layer.
Has an orange.
Yeah, near the top.
Yeah.
So I think what's happening, it's almost like the FDM tube, probably more anaerobic at the bottom of Phenol Red. So at the bottom, it has to ferment. And it's producing the acids and the gases and fill the nerve tubes. When you're at the top of that Phenol Red tube, where there's more
oxygen, it's growing faster.
It can use protein as well.
Yeah. Yes. Yeah. And maybe doing both. Maybe doing both. So think about our FTM tubes, right? It's an anaerobic environment at the bottom. Same thing with your broth tubes. If you're not shaking them, it becomes pretty anaerobic at the bottom of those tubes. There's no oxygen. So the organism is fermenting mostly at the bottom of the tube, but at the top of the tube, it could potentially be aerobic respiration. It could be using the protein, and that might be why you get that layer sometimes that forms at the top where it's actually utilizing the protein. Yep. Different type of metabolism. So this is the table from your homework from last time that we skipped over. And this is, again, is kind of a good review. So the coding system that we use, right, it was a big table in your homework from last time. So the coding we use is AG means what? Acid production from the fermentation of the sugar with gas production. And so notice if we have the base broth with the glucose added to it, so we have both protein and sugar as food choices for this organism. So we can get this result. But if we were using the base broth only, and the only food choice therefore is a protein in that tube, can you get fermentation with acid and gas production? No, no. So this would be a non-reliable result, right? Something went wrong. Somebody gave you the wrong tube or something. Somehow some type of sugar got in here. Because that's the only way you got acid and gas, right? And so what would an acid and gas tube look like? What color would it be? Yellow. And how would we know there's gas? There would be bubble in the Durham tube. So again, if we've got sugar, we can have this result, right? It means the organism fermented the glucose. We get acid and gas production. Now, if you have a tube that we code minus minus, which means no fermentation, what would this tube look like color-wise? It'd be what we started with. And what color did we kind of start with? Kind of an orangish red, right? More towards the reddish of the orange scale, right? Minus means, the second minus means that we didn't have any what? No gas production, no bubble. In this case, the other thing you want to look for is make sure that the organism grew. So Shannon asked earlier, what is this gunk in my tube? I'm like, that's your organism growing, right? And so for some of you guys, you may see some turbidity. Some of these tubes have been going for a while, so the organisms are probably dead, right? But you might see a collection of them at the bottom of your tube, right? If you don't see any growth of your organism, then you may not have gotten any growth, and that's why you're not getting any result changes, right? So in this case, it would be a viable result if you are seeing growth in your tube, okay? If you're not seeing growth in your tube, you'd repeat this test. But it is possible that when there's no protein, when there's no sugar, excuse me, and only protein, that the organism doesn't do anything. It just grows, right, and doesn't ferment the sugar or utilize the protein. So for the next one, again, with the sugar, it can do this, right? Acid and gas. We got a yellow tube, a bubble. And then when given protein only, it uses the protein. Is that possible? Absolutely, right? Because Yvonne's is a perfect example of this. Muhammad's organism is an example of this. That when given the sugar, they're going to eat that first, right? But if you give them protein only, can they eat it? Yeah, evidenced by the color changes towards the alkaline scale. It's doing something with that protein. So again, we know when we have sugar, we can get acid and gas. When you have protein, can you get acid and no gas? No. No, right? Because acid, in general too, right, you gotta have the sugar. If it's protein only and you get an acid, and it's really suspect because look it, with the sugar you got gas, and with something going on here, you don't get the gas again. That's like, okay, what's going on here? Again, something went wrong. This is not a reliable result. You should not see any acid. You should not see a yellow tube for a protein only tube. Okay, so given the choice between sugar and protein, which should they choose if they're K, and what color would they be? They ate the protein, what color would this tube be? That magenta pink color, right? So it means that they ate the protein. They broke down, degraded the peptone, the protein source. So when we give them protein only, we expect them to do the exact same thing, right? And in fact, if we coded it K because we have a pink tube, that's exactly what they did, right? So here they were given a choice, protein or sugar. They chose protein, right? Here they didn't have a choice, right? So they did the exact same thing they did earlier. They're like, okay, I'll take the protein, I'll eat that. Here again, they've given a choice, but which one did they choose? It's K, it's pink, which means they ate the protein. Okay? And then here, right, given just protein, minus, what does this tube look like? It's orange, right? There's no gas. Doesn't appear to be anything happening. This is not what we expect, right? We expect it to turn what color? We expect it to eat the protein. Given the protein and sugar, here it chose the protein. It should choose the protein again. So again, this would be suspect if you have no growth, right? Then your organism didn't grow, right? And that might be a reason why you didn't get any color change. If it did grow and you didn't get a color change, then this would not be reliable, right? Because it should have changed color. A minus means fermented the sugar, right? They chose the sugar. It's a yellow tube, no gas bubble. Okay? But then given the choice, right, between sugar and protein, they chose the sugar. Given no choice of just having protein, if it's K, it's a pink tube, which means they did what? Ate the protein. Can this happen? Yes, this can happen. So, again, given a choice, sugar and protein, they chose the sugar, right? But they didn't produce any gas, just acid. In a protein-only tube, can it be yellow with a gas bubble? Protein only? You can't ferment protein. You can't get acid and gas production. Not to mention you didn't even get gas production when you were given sugar for sure, right? So this one is suspect, right? There's something going on here, right? There's no way you should have an acid and gas production in a protein-only tube. So let's go to our scientific method, right? And so why are we performing this test? To identify bacteria that ferment glucose to acids and or gas, right? They don't always do the gas. And also to identify that the bacteria utilize protein as a carbon and energy source in the absence of a carbohydrate in the media. Okay, so how do we perform the test? We just inoculate the tubes. We let them go for 48 hours, right? Because again, the main question we're asking is, can they do this fermentation, right? And over time, they're going to run out of sugar if we let them go too long. Okay, so we take them out. So to slow some of yours down, I put them in the refrigerator to make sure that they didn't run out of sugar and start eating the protein. And then we also have the derm tube, which is going to trap any bubbles from the gas production from the fermentation. So what are our results tell us, and what are the codes being? Ag is acid with gas production, and this means they fermented the sugar, right? A minus means that acid was produced, they fermented the sugar, but no gas was produced. Neither acids nor gas were generated, and is indicated that the organism does not ferment a particular carbohydrate in the media. This would be minus, right? No acid, no gas, right? No color change from the starting point. K is another possibility, and that's if it's no fermentation at all, right? Instead, it's utilizing the protein source and it's producing alkaline products, which produces that pink color. So, what do we find out about our organisms? Is it or is it not capable of fermenting glucose, right? Does it produce gas or not? Is it not capable of doing any fermentation, but can do protein digestion? So, The other one we looked at was citrate. And this one is to determine if your organism has citrate permease. This is a membrane-bound protein that helps transport that particular sugar into the cells. So there's only one carbon source, it is the citrate, and only one nitrogen source, which can be converted and release a base. So our tubes actually started out green, right? So if you look at our uninoculated control in our rack, they started out green. Your organism will not grow, it will die if it cannot utilize the one carbon source in here. So your tube should maintain the greenish, I'm saying color because over time they'll get a little bluish. But if it's positive, there's no doubt it'll be this bright, bright blue because it will have at this point produced a lot of the ammonia byproducts, which are going to change the pH to this one to an alkaline, and for this pH indicator, Blumenthal blue, it turns blue. Okay, and you should also see growth of your organism potentially on the slant, because again, it only if it grew would it be able to produce these products, right? So again, only if it has that protein in its membrane can it take in citrate and actually grow in this media. So let's go to our scientific method. So we need to identify if the bacteria can utilize citrate as the sole carbon source, right? And it basically means, right, that they have citrate permease if they grow and produce this color change. So we inoculated, again, typically we let this one go 48 hours. This one I've let go longer because you're not going to usually get false positive results. You're just going to get nice, strong, positive results if you let it go longer. So blue color, as we said, indicates they have citrate permease and they utilize the sodium. The ammonia. They produced ammonia. Without blue color, it indicates the organism has citrate permease, allowing it to metabolize externally, but it has not yet produced alkaline environment associated with the nitrogen utilization in the media. This would term a slow-growing organism that may not produce enough to create that pH change. So this is one of those tests where, you know, sometimes you can get a false positive result if you're just looking for growth. Which is why I tell you guys to stick to looking to the color. If you look at your slant, right, and it is a greenish slant and you think you're seeing growth, the problem is sometimes that you put too much bacteria to begin with. So they didn't actually grow. This is your inoculum that you initially put on those slants. Does that make sense to you guys? Okay, so sometimes you'll get a false positive thinking that it grew when in fact you had no growth at all. We took care of the slow growers by the fact that we've let this go for a long time, right? So they should have had enough time to produce that blue color. So no growth with green color indicates they died, right? They weren't able to survive. They are negative for this test. And then catalase. So let me go to our one for this. And we definitely need to throw away those tubes that we didn't throw away last time. Okay, so we want to be able to identify that the organism can detoxify hydrogen peroxide, right? Because remember, organisms that live and especially utilize oxygen, they're going to produce these toxic forms of oxygen, one of them being hydrogen peroxide. So we can use hydrogen peroxide as a reagent and we can add it to the slide. And remember, when we did it on the slide, we have to put the bacteria right on the slide. And then we add the hydrogen peroxide. We don't want to use our loop and put it into hydrogen peroxide because the loop itself could potentially cause bubbling. And you can end up with a false positive result. And this is because of poor sensitivity of this test, right? That other things can cause this reaction, like the metal. Specificity. What did I say? Sensitivity? I did. Specificity. This is one of the few where specificity is an issue if you use the metal loop, right? And so once we add it, and then if we do it on the slant, remember we had to factor out if the organism, if the media could cause bubbling. Our media doesn't cause bubbling, so we don't get any false positive results from that. So we should see lots of bubbling, right? And why is that? What's happening? What is the hydrogen peroxide being turned into? Gas? What kind of gas? Nope. Hydrogen would be dangerous. Oxygen and water, right? So it's oxygen gas, right? Because it's in the form of a gas. That's why you get the bubbles, right? So that's the visible result. Sensitivity can be an issue, especially when we did it on the slide, where it's hard to see the bubbles, right? To see a positive test. So that's why we followed it up with doing it on the slant. And some of you guys still haven't because we didn't have enough organism. And once you put that on there, you're definitely killing them. So we'll retest later. So the organisms that exhibit catalase activity surely have an electron transport chain. This is why they have this enzyme catalase to help destroy these toxic forms of oxygen. So all your organisms, remember when we did the FTM test, all of them could grow aerobically. Some of them were strict aerobes and only grew aerobically. Some of them were facultative anaerobes where they grew at the top of your tube and could grow anaerobically without oxygen, without the electron transport chain at the bottom of your tube. But they all can grow aerobically, right? All your organisms. So all of them bubbled.
Is there one organism in the class that's actually aerotolerant?
Yeah, we have one organism that's aerotolerant, which means that it can only deal with the superoxide demutase and break down the superoxide radicals. It doesn't have catalase to break down hydrogen peroxide. But that one's not utilizing oxygen. It is an anaerobe, but is tolerant to oxygen. So we can grow it in the presence of this lab, and it's not harmed, but it's not respiring aerobically. So that one would come out negative in this test, right? It would not actually produce bubbles. And we didn't actually have anyone in this class, in this group, that had that organism. And I didn't have that organism growing to be able to show you guys that it doesn't bubble. I think there's some in the incubator today that I can show you guys, but it's not very exciting, right? It basically just doesn't bubble. Okay, so that's all the stuff that's on today's quiz too, so hopefully you were paying attention